Okay, everybody can hear me? Can everybody hear me?
Yes.
Yep. Excellent. All right, let's get started. Um, Hello again. Week 2 of this adventure in cell biology, for me at least. For you, this is week 5. So the topic that I will be talking to you about today is cell communication, which I hope is of interest. I personally enjoy it a lot. So we're going to try to make a good one out of it. In terms of lights, did we fade the? Is it okay if we do this? I think it you can see this better. All right, and this thing works. There we go. So there are a number of divisions for today's lecture, and the first part is going to be very broad principles of how cells talk to each other in many different settings. The second part is going to be focused on membrane receptors, particularly GPCRs. And then the third part is going to be about um, less, they're not necessarily less common, but they're less canonical forms of cell communication. So learning objectives, part 1, I'm again not going to read them, but they're there for your enjoyment. So to begin with, general principles, 4 forms of intercellular communication. So cells can talk to each other when they are actually touching each other, and that's what we call contact-dependent signaling. And for contact-dependent signaling, what we get is a cell that um, has a ligand that is attached to its membrane, right? I'm actually not sure if the definition of the word ligand makes any sense at this point, but basically a ligand is a molecule that is used in the communication of a signal, right? Generally speaking, a ligand is a chemical that can be shared from one cell to the next, and there are different chemical natures of ligands, etc. It's from a biochemical perspective, a ligand is a molecule that binds to something else. So in this case, there's a signaling cell. That signaling cell is sending a signal to a target cell, which is the one on the right. For that signal to be perceived, the target cell needs to have a receptor, which is the molecule that you observe there in green. So the signal molecule, or the ligand, is, in this case, attached to the membrane of the signaling cell. So the communication between these two can only really happen when the cells are touching each other. The second type of intercellular signaling is paracrine communication, or paracrine, depending on how you want to pronounce that. In this case, there's a signaling cell here at the center. The signaling cell is the one that is sending the message. And the signaling cell produces a molecule, a ligand, that in this case diffuses across the extracellular environment, and it binds to receptors on target cells that are neighboring cells, right? So, yes. Um, so the signaling molecule, this ligand, diffuses across the extracellular environment. It doesn't enter the blood. It remains in the interstitial fluid that bathes this neighborhood of cells. And so this is a relatively short distance communication. Then we have synaptic communication, in which you get basically a neuron that may have a cell body somewhere in the central nervous system. Um, and well, usually there are neurons that have cell bodies in the peripheral nervous system, but the cell body contains the nucleus of the neuron, and neurons generally have dendrites that sort of are there to perceive information, but then they have usually a single axon, which is a long um, um, protrusion of the cell of the neuron cytoplasm that basically acts as a cable, and it projects sometimes short, sometimes very, very long distances. And this neuron makes a synapse or a connection with the target cell. The two cells themselves don't really touch in this case, but there is a very, very narrow space in between the two cells. And into that space, the neuron releases neurotransmitters, which are chemicals that then bind to neurotransmitter receptors on the target cell. This is a form of long-distance communication, but it requires a relatively close interaction between the neuron, which is the signaling cell, and the target one. And finally, we get endocrine signaling, in which an endocrine cell produces hormones. And those hormones enter the bloodstream, and they are distributed all over the body via the bloodstream so that they can then reach target cells that are very distant. Oops, apologies for that. Target cells that are very distant, right? And then again, the target cell needs to have a receptor for the hormone, right? The core concept here is that the target cell, the hormone enters the bloodstream, and through there, it reaches everywhere in the body. Even though the hormones are distributed all over the body, only the cells that have receptors for that hormone can respond to the hormone, right? Again, the response is only enabled by those cells that have a receptor for that signaling molecule. Once the receptor and the signaling molecule basically get to where they need to be, right? So there's a signaling molecule that binds to a receptor. In this case, in this particular example, the receptor is bound to the membrane, and the binding of the ligand or the signaling molecule to the receptor triggers the activation of what we call a signaling cascade or a signaling pathway. And signaling pathways basically rely on events, biochemical events that take place inside of the cell, usually in a sequence. So, we have the square, the activation of the receptor activates the square protein, the square protein activates the triangle protein, the triangle activates the circle protein, and the circle protein then triggers the activation or regulates the activity, to be a little bit more precise, regulates the activity of metabolic enzymes, transcription factors, or it can rearrange the cytoskeleton in the cell. Those are called effector proteins. And the changes in the activity of the effector proteins ultimately leads to changes in cell behavior, right? We can alter the metabolism of the cell, we can alter gene expression patterns, or we can cause the cell to move, change its shape, divide, etc., right? So generally speaking, this is how signaling occurs, right? 4 forms of signaling on the left, and then the general principles of how those signals affect cells, right? We're going to go into more details about what does any of basically all of this means, and what are the specific examples that we need to pay attention to. So this table you don't need to memorize. I'm not necessarily going to ask you to remember all of these things, but it's a non-exhaustive list of hormones produced by several different tissues and glands in the body. So you see, for instance, that the anterior pituitary produces several different hormones, and those hormones tend to have one or many different target tissues. And on the rightmost column, the far right column, what you get is basically what the functional consequence of those of the action of those hormones in those tissues is. So, prolactin produced by the anterior pituitary talks to breast tissue, so to the mammary glands, and it stimulates milk production, right? So signal, target tissue, final effect. But the how the hormone causes that final effect at the cellular level, that's what we're going to try to go into in this class overall. We're going to try to give general principles of how these signals are converted into into changes in cellular behavior. Now, this is an important table, much more important than the previous one, because it basically tells you what are the general constraints to signaling pathways. Hormones can have, and signaling molecules, generally speaking, they have differences in their chemistry, right? We can actually classify them based on their chemistry. And the peptide hormones, of course, are made of amino acids. Steroid hormones are made of lipophilic they're based from cholesterol, basically. And the amine hormones are derivatives of the amino acid tyrosine. So this is where we get two categories, basically. The catecholamines, where we have norepinephrine and epinephrine, as well as dopamine, and then we have the thyroid hormones. And this is the case of hormones in particular. There are other signaling molecules that are not considered hormones that may fall in several of these categories. So on the top, you get the different categories, chemically speaking, the different categories. On the first column, you get some of their properties. So for instance, all peptide hormones have receptors located in the cell membrane. All peptide hormones activate second messenger systems. We're going to talk about what these second messenger systems are. And they act, so the target response, in other words, how this cellular change is enacted, involves the modification of existing proteins. So we're basically talking about post-translational modifications of proteins, such as phosphorylation, dephosphorylation, and a bunch of others. But phosphorylation and dephosphorylation are the ones that are more common. And at the bottom, you have just examples of those signaling molecules. Steroids, for instance, their receptors are generally in the cytoplasm or the nucleus. That kind of makes sense because steroids are able to freely diffuse across the membrane. So it doesn't really make sense for a steroid to have a receptor on the membrane of the cell, right? Because the hormone just flies through. So the receptors are located either in the cytoplasm or directly in the nucleus. There are some membrane receptors, but they're rare, and we're not really going to touch on them. And most of these, because of their nuclear localization, they tend to work as transcription factors. So they trigger changes in gene expression and thus control protein synthesis. And catecholamines they work via second messenger systems, and thyroid hormones, they tend to work by changing gene expression, because thyroid hormones are, in fact, lipophilic, mostly lipophilic. Other extracellular signaling molecules that behave kind of like hormones: prostaglandins, growth factors, cytokines, endorphins, acetylcholine are among the hydrophilic class, and all of them have cell surface receptors. Vitamin D, retinoids, and PPR ligands are hydrophobic in nature, and therefore they have intracellular receptors. Okay, so a couple of more details on these signals and their receptors and where they are and why. Cell surface receptors are transmembrane proteins, right? So that means that they have transmembrane domains. They are integral proteins to the membrane, and that means that we have basically they're stuck in the membrane. We cannot really, they're not released, right? And thus they are able to bind to hydrophilic signals that are sort of dissolved in the extracellular environment, right? Whereas intracellular receptors are located inside of the cell, sometimes in the cytoplasm, sometimes in the nucleus. They bind to hydrophobic molecules. These molecules tend to be small, so they're not particularly large in terms of their molecular size or molecular weight. Um, and because they're hydrophobic, they can actually just diffuse into the cell relatively easily. Uh, Now, for these signals to reach their target cells, they actually have to navigate the extracellular environment, which is hydrophilic. So usually hydrophobic hormones or signaling molecules are carried by proteins that kind of bind them and shield them from the aqueous environment in the extracellular fluid, right? So, when we talk about good and bad cholesterol, right? That doesn't mean that we have actual cholesterol um, elevator, like good or bad or whatever. What the good cholesterol and bad cholesterol refer to specifically are these proteins called LDL and HDL, low density and high density lipoproteins. These are proteins that bind cholesterol and other triglycerides, and they are able to carry those triglycerides and dietary fats and all that stuff through the blood, right? The blood is an aqueous environment. We don't want basically oil droplets circulating in the blood. The way that we transport them is by loading them on proteins, the carriers that basically take them from where they are incorporated to where they need to go. And that's how this signal basically happens. Sometimes there may be a handoff between an extracellular carrier and an intracellular carrier that can then bring the target molecule into the nucleus, for instance. That is also a possibility. Any questions so far? Okay. So now the ability of cells to survive and basically do what they need to do in their social context, and this is sort of important. Cells usually don't live on their own. They're social entities, right? So they are constantly exposed to several different types of signals. And in order for the cells to actually do what they are expected to do, they need to be exposed to multiple signals. So in this example, for instance, at the top, this signal here, so this cell here in the top left, is receiving three basic signals, A, B, and C. The combination of these three signals enables the cell to survive. So as long as the cell is exposed to A, B, and C, the cell will be able to survive. But if on top of A, B, and C, you add two other signals, D and E, in the presence of the survival cells that basically provide you with this baseline, D and E cause the cell to divide, right? But now, if instead of D and E, you put F and G, so two different signals, then the cell is not going to simply survive. It's going to differentiate, right? So we can start with say, a cell from I don't know how much developmental biology you know, but let's say a cell from the endoderm, which is one of the three primordial layers in embryonic development. The presence of these signals would enable that cell from the endoderm to initiate their differentiation into, say, um, cells of the gut, for instance, right? So cells of the liver or whatever. In the absence of these signals, the cell will die, right? Because the survival cells are not there, right? You could expose these cells to either D and E or F and G, and probably neither of those the cell would not be able to divide or differentiate because the survival cells are not present, all right? Now, another very important concept is that the signal or the effect of a particular signaling molecule depends on the receptor that is found on the the type of receptor and the type of cell that this cell is that this signal is basically activating, right? So, for instance, in this particular example, we have a single type of signal, acetylcholine. It's a small molecule that is very, very well known and very well studied. This is the main neurotransmitter from the parasympathetic nervous system, which is one of the branches of the autonomic nervous system. So a bunch of things depend on acetylcholine. When you put acetylcholine on the cells of the heart, but not on any cell of the heart, in the pacemaker cells of the heart, right? The heart has pacemaker cells and contractile cells. Acetylcholine doesn't do anything on the contractile cells because there are no good receptors for acetylcholine. But in the pacemaker cells, there's a specific type of receptor that binds to acetylcholine, and it triggers the decrease in the rate of firing of the heart, right? If you are taking physio, next week I'm going to tell you how that happens. If you are not taking physio, I suggest that you take it next year. In the salivary glands is a completely different type of cell, and it doesn't have the same functions, right? But the same molecule binds to a similar receptor. And in this case here, the salivary cell doesn't really control the pacing of the heart, but it does have a lot of secretions. So binding of acetylcholine to its receptor causes the cell to secrete stuff, right? In skeletal muscle cells, acetylcholine is the main neurotransmitter that mediates muscle contraction, skeletal muscle contraction. And here, acetylcholine binds to a completely different receptor, which is what we call the nicotinic acetylcholine receptor. The nicotinic acetylcholine receptor is a ligand-gated ion channel. So binding of, in this case, two molecules of acetylcholine to the receptor triggers the opening of the channel, and this triggers stimulation of the muscle cell and thus contraction. Another example of this, and I think it's actually a more interesting example, that is related precisely to this, the fact that the type of receptor determines the function. If you have a smooth muscle cell, and the smooth muscle cell has alpha adrenergic receptors. Adrenergic receptors are the ones that bind to epinephrine and norepinephrine. So the same epinephrine or norepinephrine can cause contraction or relaxation of the muscle depending on the type of receptor. If the smooth muscle cell has alpha receptors, the norepinephrine will cause contraction. But if the cell has beta receptors, the same signal will cause relaxation. And that happens because the two different receptors are associated with different intracellular signaling pathways. All right? So well, no questions so far. Any questions, by the way? Okay. So the timing of the responses is also different, right? If you have a signaling molecule that binds to the receptor and the receptor triggers an intracellular signaling pathway that operates via altering protein function by phosphorylation and all those things that I mentioned a little earlier, this response will tend to be fast because it depends on very direct and short-lived interactions between proteins inside of the cell. But if we have a receptor that operates by going into the nucleus and changing patterns of gene expression that depend on protein synthesis, all of that takes much longer, right? So, and this is the case for some extracellular signals that bind to cell surface receptors. It's the case for most signals that bind to intracellular receptors because all of them have to go into the nucleus. The change in cellular behavior mediated by intracellular signaling pathways can be very fast, sometimes less than a second, right? Quite often will be just a few seconds into just a few minutes, but responses that depend on protein synthesis and gene expression changes will take dozens of minutes to several hours, right? So these are two very different time scales, right? Now, I'm going to tell you a couple of things about the main classes of cell surface receptors. There are receptors that operate as ion channels, right? And I don't know how much about ion channels you know, but these are basically proteins that are present on the membrane, and they form a pathway for charged particles, ions, to move across the membrane, either from the outside to the inside or from the inside to the outside, right? So ion channels come in several different flavors. And one of the flavors of ion channels is what we call ligand-gated ion channels, or in this case, since we're talking about this from the perspective of signaling, these are ion channel coupled receptors. In blue here, you have several different ions. Most of them are cations. So we understand a lot better what happens with positively charged ions than what happens with negatively charged ones. Also, for the purpose of permeation across the membrane, positive ions such as sodium, potassium, and calcium, they tend to be more frequently utilized by the cell. The two ions that are negatively charged that are really important in physiology are chloride and bicarbonate. And there are channels there are definitely channels for chloride. There are transporters for bicarbonate. Anyway, the ion channel is closed in the left side of this figure, right? So the ions cannot go through. But when the signaling molecule binds to its binding site on this particular receptor, that causes a conformational change in the protein that leads to the opening of this passage, and thus we can have ions move through. The other major class of cell surface receptors, and we are going to spend a decent amount of time on them on the second part of the class, are the G protein coupled receptors. G protein coupled receptors are composed of a 7 transmembrane receptor that has it's basically a protein that traverses the membrane 7 times. It has an extracellular amino terminal, so the first amino acid in the protein is outside of the cell. The C terminus is intracellular, and this is the protein that will actually bind to the signaling molecule. The binding site will usually be somewhere in the extracellular environment. The other component of these GPCRs is an inactive G protein. The G protein is a complex, what we call a heterotrimer, 3 different polypeptides. There is an alpha subunit, a beta subunit, and a gamma subunit. The alpha is the one that we're going to be talking the most about. And this G protein, this heterotrimeric G protein, it's called G protein because they bind to GTP. So in other words, it's a GTP binding protein. The inactive G protein lives somewhere close to the receptor. And as part of the system, there's also some kind of enzyme, right? This enzyme can be several different types of things. When we put the ligand will bind to the receptor itself, and the receptor will the receptor will become activated by the binding of the signal molecule. And when that activation happens, the receptor is able to directly interact with the G protein, and it triggers its activation, right? And then the activated G protein can actually lead to the activation of this enzyme element of the system, and then the enzyme will do a bunch of other things. And then the heterotrimer can actually dissociate into the alpha subunit, which is the one in darker, well, this is greenish. And the beta gamma subunits, they also have signaling properties, but they act independently of the alpha subunit, right? So again, the G protein, the 7-transmembrane receptor, the GTP binding protein that has alpha, beta, and gamma, and then we have this enzyme. So activation of the receptor leads to activation of the G protein. The G protein can then, the alpha subunit activates the enzyme. The beta gamma subunit does a bunch of other things that we are not really going to go into. The third important class of receptors are the enzyme-coupled receptors. Enzyme-coupled receptors are monomeric receptor proteins that are a single transmembrane pass. So they have only one segment that crosses the membrane. And they have an outside, an extracellular ligand binding domain and an intracellular catalytic domain. This is where the enzyme is, right? The enzyme function, the enzymatic function is. When the signal molecule binds to the receptors dimerize. So initially we have inactive receptors are found as monomers. When the ligand comes in, the receptor dimerizes, right? And this dimerization triggers the activation of the catalytic domain. The catalytic domain is a kinase, for the most part, is a kinase domain. So this catalytic domain causes the phosphorylation of proteins. Interestingly, what really happens here is that let's call it left and right. The right half of the receptor cross-phosphorylates the left hand of the receptor, and the left hand of the receptor cross-phosphorylates the right side of the receptor, right? So there's this sort of, in some textbooks, you will read this as autophosphorylation, right? The self-phosphorylation of the receptor. But it's basically a cross, it's like a handshake of sorts. And then the active catalytic domain that has been phosphorylated does a bunch of other things that we are going to discuss a little later. The other possibility with these enzyme-coupled receptors is that the receptor continues to be monomeric in its inactive form. The presence of the signaling molecule causes the receptor to dimerize, but the dimerization and the activation of the receptor triggers the activation of an associated enzyme. So in this case, the receptor itself doesn't have enzymatic activity, but it's closely associated with an enzyme after it becomes activated. Does that make sense? So these are basically two different flavors of a similar process, right? Any questions? Any questions? Any questions? Nope. Okay. So what characterizes all of these receptors is the fact that they have to dimerize, right? The base receptor of sorts is found in its inactive conformation. The two halves of the receptor are separate from each other, but the binding of the signal causes them to pair up, right? That's the key characteristic of these enzymes, of these receptors. All right. Now we've said a couple of times that these receptors work by relaying signals intracellularly using intracellular signaling molecules, right? This relay can happen in multiple different ways. In this particular example, what we have is that the signal is relayed by an event of phosphorylation, which basically involves a protein kinase, right? Kinases are, you've heard about kinases before, right? So kinases are enzymes that are able to transfer phosphate groups from ATP to a target protein. So when the signal is in, so again, the signal binds to the receptor activated that G protein, for instance, and that activation process leads to the turning on of a protein kinase, right? So activated receptor triggers the activation of this kinase. The kinase takes a molecule of ATP and it transfers the phosphate group, the last phosphate group from that ATP, onto a target protein, right? And this phosphorylated protein becomes active, and then it can relay the signal downstream. This activation is never, it's never forever, right? So this signal has to be turned off for, at some point. And the signal to the enzyme that helps turn this process off is called a phosphatase. Phosphatases recognize phosphate groups on their target proteins, and they basically slice that phosphate group off. So they remove the phosphate group, and when the phosphate group is removed, this protein in particular is no longer phosphorylated, and it becomes inactive. Okay? That's the signal by phosphorylation. Other proteins or other signaling molecules become active when they bind to GTP, right? This is a relatively complicated thing to understand because it needs a few additional partners, right? When the signal comes in, the signal triggers a protein that's called a guanine exchange factor, or GEF. The GEF observes that this, the protein that we're interested in is bound to GDP initially. This GDP is similar to ADP, but instead of being an adenine, it's a guanine nucleotide, right? And when this protein is bound to GDP, it's inactive, right? So there's nothing really going on. The GDP sort of is usually a marker of inactivity. But when the signal comes in from the outside, this guanine exchange factor will trigger the exchange of GDP for GTP, right? GDP comes out, GTP comes in. And now this protein is bound to GTP, and GTP is a marker of activity, not inactivity, right? The GTP bound form is active. And this activated switch can then relay the signal further. The termination of this process happens with GTP hydrolysis. So for a number of different reasons, there are factors that control the ability of this signaling molecule to hydrolyze the GTP that they have acquired. So the hydrolysis of this GTP triggers the release of a phosphate group, and then the protein is back to its GDP inactive form, right? So this is a cycle, right? This signaling protein cycles between the inactive GDP bound form to the GTP active form, right? And back and forth, back and forth. And there are factors that regulate that process. As I said before, the proteins that regulate this transition are called guanine exchange factors. The exchange is because, again, GDP is bound to is the inactive, and it gets exchanged. So GDP comes out, and it's exchanged by a GTP molecule. GTP binding causes this GTPase to become activated. And the return to the inactive state is usually mediated by a GAP protein. GTPase activating protein. That's what GAP stands for. So GTPase activating proteins interact with the GTPases. They turn on their GTPase activity, and the GTPase can then hydrolyze that GTP into GDP, becoming inactive in the process. Okay? Does that make sense? This is a relatively esoteric thing that, in my experience, sometimes it's a little harder to internalize. Any questions? Okay. Now, intracellular receptors. Sorry, these intracellular signals are usually generated in complexes that involve the receptor and a bunch of different signaling proteins. So in this particular example, for instance, you have the inactive receptor that's bound to the membrane, and there's a little tail of the receptor that interacts with the scaffolding protein, or a scaffold, right? And the scaffold protein is a relatively large protein that is able to recruit and bind to other proteins. So in other words, it's like a protein that helps recruit things that kind of act in the same pathway. So this scaffold is bound to 3 different proteins, protein 1, 2, and 3. And all of them are part of the same signaling pathway. They, in fact, tend to be activated in a sequence. First 1, first 2, first 3. So when the signal molecule arrives, the receptor becomes activated. And when the receptor becomes activated, then the receptor activates protein 1, which happens to be nearby. And protein 1 activates protein 2, which also happens to be nearby. And protein 2 activates protein 3, so on and so forth. This would be much less likely to happen if these proteins were dispersed diffusely over the cytoplasm of the cell. But the fact that there is a scaffold protein brings them all together, and now the signaling process is made all the more efficient, right? The other thing that can happen is the assembly of these signaling complexes at the receptor itself, right? This is actually what happens with a specific type of receptor called the receptor tyrosine kinase. The activation of the signal molecule activates the receptor becomes phosphorylated. But the phosphorylation of the receptor creates docking sites for several signaling proteins, in this case, proteins 1, 2, and 3. So if the receptor is not phosphorylated, which is when the receptor is inactive, these proteins cannot bind to the receptor. But when the receptor becomes activated and then phosphorylated, these signaling proteins can dock on the receptor, and they can become active and then trigger further signaling events downstream. Finally, the docking can occur at the membrane itself, right? Because remember that the membrane of the cell is made of phospholipids, right? And the distribution of these phospholipids is not homogeneous, right? There are some specific phospholipids that are more concentrated in the face of the membrane that is on the side of the membrane that is facing the cytoplasm, right? Some of those phospholipids are called phosphoinositides, and they are phosphorylated phospholipids, right? And in the absence of the ligand, the receptor to that ligand is just hanging there, and the intracellular signaling proteins are also sort of somewhere, right? But when the signal binds to the receptor becomes activated, and the enzyme associated with this receptor triggers the phosphorylation of these phospholipids, right? So these phospholipids become what we call hyperphosphorylated. This means that instead of being phosphorylated two times, now they're phosphorylated 3 times. Or if they are phosphorylated 3, they can be phosphorylated 4, whatever. Regardless, what matters is that now these hyperphosphorylated sites on the membrane can be recognized by these signaling proteins that then bind and become activated, and the signaling cascade operates in that way. So activation of the receptor triggers phosphorylation of these phospholipids, binding or docking of protein 1, binding and docking of protein 2 nearby, and this proximity enables 1 to activate 2, right? And the protein 2 then triggers the further signaling downstream. Does this make any sense? Any questions about this? All right. A relatively important concept in this entire topic is the fact that the concentration of extracellular signals tends to be very, very, very low. Um, by low, I mean we're talking about nano to picomolar in the plasma, right? So these are extremely, extremely low concentrations. The way that signaling processes work is by amplifying these signals intracellularly. So the idea is that you have a ligand bound to the receptor. The receptor activates an enzyme, and this enzyme has the ability to multiply the effect of that single signal by making multiple copies of second messenger molecules and activating thousands of copies of effector intracellular proteins. To put it in perspective, in the eye, there's the receptor for light in the retina of the eye is called rhodopsin. And rhodopsin is able to be excited by one photon hitting that rhodopsin molecule, right? The activation of one rhodopsin molecule triggers the activation of 500 G proteins, right? The activation of 500 G proteins triggers the activation of 500 cyclic GMP phosphodiesterases, which are enzymes that hydrolyze cyclic GMP. The activation of these 500 cyclic GMP phosphodiesterases leads to the hydrolysis, or basically the destruction, of 10 to the fifth molecules of cyclic GMP. That's 100,000 molecules of cyclic GMP. The hydrolysis of 100,000 molecules of cyclic GMP triggers the closure of 250 cation channels. The closure of those 250 cation channels prevents the entry of between 1 and 10 million sodium ions into the cell for a period of about 1 second. And this prevents the fact that there is no longer entry of this amount of sodium triggers the change in the membrane potential of this retinal cell by 1 millivolt, right? So one photon, one particle of light triggers the hydrolysis of 100,000 molecules of cyclic GMP, and they prevent the entry of up to 10 million sodium ions into these cells, right? So as this happens over time, and probably millions of molecules of rhodopsin are being activated every second or so, what you get is relatively large changes in membrane potential, and that's what ultimately triggers the activation of neurons, and that relays the signal to the brain, and that's how we see, right? So this gives you sort of an idea of how the cells are able to amplify low levels of extracellular signal to relatively strong intracellular responses. This table is interesting. I will go over a couple of details here. These are what we call the second messenger pathways. It is an expectation that you will know these, all right? So in the column basically has second messengers, the molecule that they're synthesized from, the enzyme that makes them, the type of receptor that they activate, or the type of intermediate signal that they are associated with, the cellular action, and the effects on the cell. So second messengers can be nucleotides, and there are two general nucleotides that are used in this process. Cyclic AMP, which is derived from ATP, and cyclic GMP, which is derived from GTP. The enzymes that make them are called cyclases. So in the case of cyclic AMP, adenylyl cyclase makes cyclic AMP from ATP, and it is activated by a specific family of G protein coupled receptors. And this cyclic AMP, when it is produced, it triggers the activation of protein kinases, particularly a protein kinase called PKA. And this is not the only action of cyclic AMP, but it's the most and best. It's the most studied and the best characterized. And when PKA becomes activated, that triggers phosphorylation of proteins, right? And the phosphorylation of proteins changes their activity, and a bunch of different things can happen in the cell. Cyclic GMP is made from GTP by two forms of a guanylyl cyclase, right? There is a membrane-bound guanylyl cyclase that is, again, stuck in the membrane, and it triggers protein. This cyclic GMP triggers protein kinases, and again, protein kinases phosphorylate target proteins. There's another version of guanylyl cyclase, which is the soluble guanylyl cyclase. This enzyme is found in the cytosol of cells. It's not in the membrane. And this guanylyl cyclase is the receptor for a gas hormone called nitric oxide. This nitric oxide is extremely important in the functioning of the cardiovascular system, among a bunch of different things. And cyclic GMP, so nitric oxide activates guanylyl cyclase. Guanylyl cyclase makes cyclic GMP. This cyclic GMP will bind to ion channels and a bunch of different things and trigger changes in cell behavior that way. The second class of second messengers is derived from lipids, and there are two important ones here. IP3, inositol triphosphate, and then DAG, or diacylglycerol. Both of them are derived from membrane phospholipids, particularly a phospholipid called PIP2, phosphoinositol diphosphate. I remember, but PIP2. PIP2 is attacked by an amplifier enzyme called phospholipase C. Phospholipase C basically grabs PIP2 and it splits it into IP3 and diacylglycerol. Diacylglycerol stays stuck on the membrane, and it triggers the activation. It becomes a docking site and activating signal for a different type of kinase called protein kinase C, or PKC. IP3 becomes soluble, and it is released from the membrane into the cytosol, and it binds to its own receptors that are present usually in the they're present usually in the membrane of the endoplasmic reticulum. That IP3 receptor is basically an ion channel. It's a calcium channel. So when IP3 binds to that receptor, it triggers the release of calcium from the endoplasmic reticulum, and then it does the things that calcium does. Calcium is considered a second messenger on its own merit, and depending on the cell type, calcium can bind to calmodulin, which is an intracellular calcium sensor, and the complex made by calcium and calmodulin can, in fact, alter the activity of several different enzymes inside of the cell. Calmodulin is not the only calcium sensor in the cell. There are several other calcium-sensitive proteins in the cell, but they're beyond the scope of our class, and you will see them if that's necessary. All you need to know at this point is that calmodulin and these other proteins are able to bind calcium, and when they bind calcium, they trigger activation of other downstream signals, and then the cell changes its activity. Does that make sense? Any questions? Cool. Again, it is an expectation that you will know this table. Now, cells can adjust their sensitivities to signals, right? You may have a constant level of a particular signal, and if you think from if you think about this from an addiction perspective, for instance, like the biology of addiction, usually what happens when addiction, addiction develops, addiction is the sort of the last step of a protein that basically, of a process that leads to the buildup of tolerance to a particular signal, right? So initially, when you first expose the body to a particular drug, um, the body is usually maximally sensitive, and low amounts of the drug can lead to strong effects. But the more exposure you get, the higher the doses you need in order to achieve the same effect, right? So you go increasing the dose, um, and as you increase the dose, the cell becomes less and less sensitive, basically, to the drug. So there are some mechanisms are mediated by changes in the receptors, right? So quite often what happens is that the receptors can be sequestered. So the presence of the signal at the receptor becomes activated, and after a certain period of time, the receptor can be endocytosed, so it can be basically loaded into an endosome, and the cell basically catches that the cell catches that little vesicle and holds onto it for a certain period of time, and then the vesicle can actually be sent back to the membrane, and the receptor can be recycled. The receptors can also be downregulated, right? So binding of the binding of the ligand to the receptor triggers the endocytosis of the receptor with the ligand, but instead of being recycled back to the membrane, this vesicle that contains the receptor is destined to the lysosome, where the cell will basically destroy the receptor and the ligand, right? That reduces the amount of receptors in the that are present on the membrane of the cell. And there are signaling mechanisms that can lead to the inactivation of the receptor. So binding of the ligand triggers a signaling pathway that, among all of the things that the pathway does, it triggers an event that inactivates the receptor and basically prevents the receptor from continuing to signal, right? So those are receptor-mediated mechanisms of sensitization or desensitization in this case. Some other mechanisms are mediated by the signals. So in this case, for instance, the activation of the receptor triggers the activation of an intracellular signaling protein. The intracellular signaling protein will activate a downstream signaling cascade, but as part of that, one of the downstream elements of the pathway triggers the inactivation of this sort of master signaling mechanism, right? And the other thing that happens is that it's sort of a similar situation. The activated signaling protein. So again, the receptor binds to the ligand, the receptor becomes activated, and then the intracellular pathway is activated. The activation of the pathway leads to a slightly more convoluted process by which an inhibitory protein is produced. And this inhibitory protein can then basically block the interaction between the receptor and the signaling molecule, and that basically shuts off the process. It's like a circuit block in a way, right? Any questions about this? No? Anything at home? Okay. Um, so what about we take a 5-minute break? Yeah, because it's 7 o'clock. It's about time for a break. Yeah. Okay. Let's take 5. All right. So the we've talked quite a bit about the general idea of membrane-bound receptors, right? But let's not forget about the nuclear ones. Most nuclear receptors actually share a relatively common activation mechanism. On the left side of this figure, what you get are basically the general structures of several nuclear receptors. The cortisol receptor, estrogen, progesterone, vitamin D receptors, all of these, thyroid hormone receptor, retinoic acid, all of these are hormones that are hydrophobic. And the length of the line basically represents the length of the amino acid sequence. But what all of them have in common is the presence of a DNA binding domain. That DNA binding domain is represented in the light green shaded part of the figure at the center. The idea is that when the receptor is in its inactive form, the receptor is basically kept in an inhibitory conformation by a chaperone protein, right? So this inhibitory protein is basically a protein that binds to this inactive receptor and it keeps it in that inactive conformation. The receptor itself contains a binding site for the inhibitory protein. It contains a region that is able to bind to the ligand. It contains the DNA binding domain, which, as the name sort of implies, it's able to bind directly to the DNA. And then it has a transcription activating domain or a transactivating domain, where this is the part of the protein that will interact with other transcription regulators, right? So the idea is that when the ligand is present, so somehow this ligand makes it all the way into the nucleus where this inactive receptor is, the ligand basically displaces the inhibitory protein. You see that the inhibitory protein is not there anymore. And the binding of the ligand causes a conformational change on this receptor that exposes the DNA binding domain. It enables it to bind to a receptor binding element, which is a DNA sequence in the nucleus of the cell, usually located in the promoter region of a particular gene. Not always, but usually there. And this enables the transactivating domain to bind to these co-activator proteins. And this complex sits on the promoter of the gene, and it is able to recruit the transcription machinery. It brings the RNA polymerase and transcription initiation factors 1, 2, and 3, and 700. And this basically helps put the polymerase where it needs to sit in order to transcribe the DNA into a molecule of RNA, and then we express new genes, right? And all of these receptors basically work in this exact same way. The differences are what kind of co-activators they need, what kind of inhibitory chaperones they have, and that's basically it, right? Oops. Now, the response to the nuclear receptors can be biphasic. What does this mean? You have a steroid, right? A steroid hormone, think of estrogen, right? Or any of these examples. And its receptor. So when the steroid hormone binds to this receptor, what we get is the activation of primary response genes. So you see here we have a molecule of DNA, and the yellow shaded regions represent different genes, right? So the hormone receptor only is able to activate these primary response genes. So the early primary response leads to the production of these proteins here, the circles and the squares. Those initially primary response genes can then inhibit themselves after this is sort of a negative feedback situation where when these things have been produced in enough quantities, that prevents them from continuing to be produced. But then they also serve as transcription factors themselves. So they can then bind to different target genes, and that leads to the production of secondary response proteins. And the secondary response proteins can then lead to coordinated changes in cell behavior. So you have it's a biphasic response because you first have a wave of early primary response, and then you have a wave of secondary response proteins. This is basically how nuclear receptors can amplify their signals, right? The same way that in membrane receptors, the signal amplification happens by producing many molecules of second messengers. Here, what you get is the production of the early primary response genes, and these early primary response genes then can trigger the activity, or they can trigger the expression of several other genes. So here, your primary response genes are sort of the second messengers, right? Or they are analogous to the second messengers in the previous examples. Now, there are three types of nuclear receptors. This is more than anything for your information, but basically, type 1 receptors, they are located in the cytoplasm or in the nucleus, and they trigger the activation of this type of complex. You don't need to remember the details. This is nothing you need to remember in detail. Something that characterizes them is that they are accompanied by a chaperone called HSP90. The HSP90 is able to help bring the receptor into the nucleus. Type 2A receptors are located in the nucleus. They are not present in the cytoplasm. And they trigger transcriptional activator. They tend to heterodimerize, right? So as the type 1 receptors homodimerize, so two molecules of receptor trigger the activity. Here, two molecules of different receptors. So here you have the RXR is one type of receptor, the other is a nuclear receptor, whatever, and they homodimerize, and they do what they do. And type B are called orphan receptors because they are known to be homodimers. Sometimes they are monomers. But what's important here is that we know that they exist, but we don't know who activates them, right? So there's active research in understanding what are the pathways and how is it that these receptors become activated, right? And that's it for the first part. So what I'm going to do is we're just going to go straight into the second part, because why not? I tend to, one of the things that I tend to do is just sort of pause the recording and restart it, but that's what I do in my courses. This is not my course. So anyway, part 2 is signaling pathways activated by cell surface receptors. So we're going to talk quite a bit more about GPCRs and, in some cases, receptor tyrosine kinases. So you can ignore that for now. And then these are your learning objectives. And we're going to start by remembering that GPCRs are able to relay extracellular signals, and they use heterotrimeric G proteins to do that, right? The heterotrimeric G protein is basically made of an alpha, a beta, and a gamma subunits. The alpha and gamma subunits are attached to the membrane by prenylation, which is a post-translation modification that enables them to basically stick or sort of hook onto the membrane without being transmembrane themselves. So these are membrane-associated proteins, but they're not integral proteins. And the alpha subunit is the one that is able to bind to GDP or GTP, right? So the GTPase part of this heterotrimeric G protein is in the alpha subunit. In the lower figure, what you get is basically a ribbon structure of our understanding of the crystal structures of these proteins, where this element here is the receptor. The ligand binding pocket is located in the extracellular side, but it's buried a little deeper into the core of the receptor. And the receptor has this little sort of arm that it is able to directly interact with the alpha subunit, which you see here in this sort of teal color. And right there at the center, that's the GDP, the nucleotide binding pocket, basically. And then you have beta and gamma subunits. The beta has a lot of beta-pleated sheets. The gamma subunit is a little smaller. Now, in the inactive conformation, the heterotrimeric G protein is sort of separate from the receptor, and it's bound to GDP. When the signal molecule arrives, the receptor becomes activated, and the alpha subunit directly interacts with the receptor. This direct interaction causes a conformational change that basically kicks the GDP out, and it enables the binding of GTP. So this is a nucleotide exchange or a guanine exchange process. And the binding of GTP triggers the activation of the alpha subunit, which on its own, it's going to cause cellular changes. The beta gamma subunit dissociates from the alpha subunit, and it can trigger activation of effector proteins on its own. This is an important table as well. This is a table that tells you basically some of the most important family members of the G protein coupled receptors. Family 1 contains the GS member. The GS is chiefly mediated by the activity of the alpha subunit, right? The GS subunit activates an enzyme called adenylyl cyclase, and it triggers the production of cyclic AMP. The GS can also activate calcium channels. The GOLF subunit, the alpha subunit of the GOLF protein, does a similar thing, but specifically in neurons of the olfactory system. So this is one of the things that characterizes G protein coupled receptors is that there are roughly, there are somewhere between 800 and 1,000 different genes that encode for G protein coupled receptors. So with alternate splicing and all sorts of molecular tricks, the body can actually have thousands and thousands and thousands of versions of these proteins. And our ability to smell, the entirety of our olfactory system is based on these G protein coupled receptors, right? So every odorant receptor, every molecule that we can smell because there is one specific type of G protein coupled receptor. Family 2, the most important member is the GI protein. Here, the alpha subunit is basically sort of opposite to the alpha S, right? So the GS protein activates adenylyl cyclase. The GI inhibits adenylyl cyclase, and this is mediated by the alpha subunit. The beta gamma subunit from these GI proteins can actually signal on its own by activating potassium channels. And the activation of potassium channels has profound consequences, particularly for excitable cells, because the activation of potassium channels triggers the hyperpolarization of the cell. And usually this means that cells become more, they become less excitable. So it's sort of a way to turn the cells off. Another member of family 2 is the GO. And the GO proteins are primarily act via the beta gamma subunit that leads also to the activation of potassium channels. And both the alpha and beta gamma subunit can activate phospholipase C beta, which is an important enzyme in several different signaling processes. And then there's transducin, which is the G protein that's in rod photoreceptors and it mediates a significant component of our vision. And family 3, the key element here is GQ. GQ via activating the alpha subunit triggers the activation of phospholipase C beta. We're going to talk about this pathway quite a bit. And then there is family 4 that includes the G12-13 proteins, which can activate a specific family of small GTPases called Rho, right? And Rho is a type of GTPase that's heavily involved in the regulation of the cytoskeleton. So activation of G12-13 triggers changes in cytoskeletal activity, and we can trigger cell migration, the change of cell shape, so on and so forth. You may notice that in this table there are 3 elements that are marked with an asterisk. Those are the ones that you absolutely have to know, right? The other ones are, it's a good idea to know all of them, but the minimum you need to know is GS, GI, and GQ. Okay? Any questions so far? Okay. So GS and GI, both of them they involve adenylyl cyclase. So the adenylyl cyclase enzyme, which is depicted by this little box there, it basically acts on ATP. ATP, your standard nucleotide triphosphate. What this enzyme does is basically it creates a bond between it hydrolyzes 2 phosphates out of ATP, and it basically is able to bend the molecule such that there is a double linkage between the ribose sugar and the phosphate group. So there's basically there's a sort of a ring or a cycle of covalent bonds between the ribose and the phosphate, right? So this molecule here is what we call cyclic AMP. So it's adenosine monophosphate, but it's cyclic because again, the phosphate is linked twice to the ribose. And then this cyclic AMP molecule can be hydrolyzed back into naked or whatever standard AMP, adenosine monophosphate, by enzymes that are called phosphodiesterases. The phosphodiesterase will basically break that bond there and turn this cyclic AMP into regular AMP, right? So again, you have an enzyme that takes ATP and turns it into cyclic AMP. That is the adenylyl cyclase. There is an enzyme that takes cyclic AMP and inactivates it into AMP. That is the cyclic AMP phosphodiesterase. GS coupled receptors, so the alpha subunit of GS coupled protein receptors activates adenylyl cyclase. So it increases the conversion of ATP into cyclic AMP. GI, so the alpha subunit in GI proteins inhibits adenylyl cyclase, and thus we get less cyclic AMP production, right? That's how we regulate the system. Cyclic AMP is the one that has the cellular effects. And the termination of the response is mediated by the phosphodiesterase. So this table basically tells you a limited number of hormones that actually act via increasing levels of cyclic AMP. So the TSH, the thyroid stimulating hormone, on the thyroid gland triggers synthesis of thyroid hormones. In muscle cells, adrenaline triggers glycogen breakdown. In the heart, adrenaline triggers increase in heart rate and the force of contraction of the heart, et cetera, et cetera, et cetera. This is a table that I don't necessarily think you need to memorize by heart, but it's to put context to why we're talking about this. This is important. Now, this is supposed to come at the end, but since it's there, cyclic AMP has three main effects. The first that we're going to mention, and it's not the most important, but it is very important in some settings, is the activation of cyclic nucleotide gated ion channels, right? So you have the receptor binds to the signaling molecule. We trigger the activation of the G protein, activation of adenylyl cyclase, production of cyclic AMP. And this cyclic AMP can directly open a type of channel called CNG that is basically permeable to calcium and sodium. And then the calcium that comes into the cell through this channel has a bunch of different effects. This is one of the ways in which we are able to smell, because again, you see that this is the G olfactory receptor. Now, the canonical type of signaling involves the activation of protein kinase A. So the signal binds to the receptor activates adenylyl cyclase. So the receptor activates the G protein, in this case, GS. And GS activates adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. And cyclic AMP then binds to the regulatory subunit of an inactive protein kinase A molecule. pKa in its inactive state is composed of 4 subunits, 2 regulatory in gray, 2 catalytic in blue. The cyclic AMP binds to the regulatory subunits, and that causes the dissociation of the regulatory subunits from the catalytic subunits. And the catalytic subunits then become activated when this happens. The catalytic subunits are the ones with with kinase activity, and they're the ones that are going to trigger the phosphorylation of downstream proteins. And the third form of cyclic AMP signaling is mediated by a guanine exchange factor called EPAC1. So this is one example mediated by beta-2 adrenergic receptors. They are coupled to GS. GS activates adenylyl cyclase. We get cyclic AMP production, and cyclic AMP triggers the activation of EPAC1. EPAC1 is a guanine exchange factor for a small monomeric GTPase called RAP. So RAP is one of these proteins that it's inactive when it's bound to GDP. It's active when it's bound to GTP. So that the switch from GDP bound to GTP bound is catalyzed by EPAC1. So increases in cyclic AMP trigger activate EPAC1. EPAC1 triggers activation of RAP1, and then things happen in the cell, primarily associated with intercellular junctions and regulation of the cytoskeleton. So 3 forms of signal transduction mediated by cyclic AMP. These are the 3. The canonical one, the one that everybody talks about, and the one that you absolutely have to know is the canonical one. I'd be very happy if you, because my research involves some EPAC related stuff, so I'm partial to it. The EPAC one and then cyclic nucleotide gated channels are also relevant. Any questions so far? Okay. What happens when you activate pKa? Because again, that's the minimum thing that I'm asking you to know. What happens when pKa becomes active? pKa has a nuclear localization signal. So the activated pKa can actually cross the nuclear pore and enter the nucleus where it can phosphorylate a transcription factor called CREB, cyclic AMP, cyclic AMP response element binding protein, basically. So when pKa phosphorylates CREB, CREB can then bind to a region in the promoter of target genes called cyclic AMP response element, or CRE. And the phosphorylation of CREB creates a binding site for a partner protein called CREB binding protein, or CBP. Not the CBP that we're not going to talk about, but this complex basically binds to the DNA, and it triggers DNA gene expression, right? So we trigger transcription of genes. Now, that's the easy part, right? This figure shows you that pKa has a ton of different things going on for itself. I am not asking you to remember this graph, but I do want to convey the very important point that these signaling proteins form a network of things that is extremely complex, very pleiotropic, as the old school scientists would say, and many things can happen. And these things are usually cell type specific, context dependent, ligand dependent, right? So two different ligands may trigger the activation in the same cell of two different branches of these of all of these pathways. And what really drives what drives which pathway gets activated is which pairs of proteins are activated together, who interacts with whom, so on and so forth. So all I want, again, the central elements are the receptors, the adenylyl cyclase right here, production of cyclic AMP. So this cyclic AMP will bind and activate pKa, and pKa has all of these other things. Tucked into the figure is also EPAC, and there are a bunch of other things that, again, serves the point of highlighting how interconnected all of these things are. Okay? Any questions? No? Okay. If anything, this side of the figure is really important. Okay, that's about cyclic AMP. What happens with phospholipids on the membrane? This is where the GQ pathway becomes important. So this gray band here represents the the cytoplasmic phase of the plasma membrane. So it's one of the two monolayers of the plasma membrane. So embedded in the phospholipid bilayer, what we get in this inner or cytoplasmic phase of the plasma membrane, we have these phospholipids that are, again, embedded in the membrane, and some of them are this PIP2, right? It's a phosphatidyl inositol. That's what PI stands for. And then this is biphosphate, right? So there are two phosphate molecules that are important here. The phosphate bound to carbon number 4 in the inositol sugar, and then the phosphate bound to carbon number 5, right? The third phosphate here is the structural phosphate of the phospholipid, right? Remember that all phospholipids have at least one phosphate group that connects the glycerol to the rest of the phospholipid head. So again, phosphatidyl inositol biphosphate. There's an enzyme called phospholipase C beta that basically hydrolyzes that bond right there, right? And it releases the inositol molecule bound to 3 phosphate groups. So this is called inositol 1,4,5 trisphosphate, or triphosphate, IP3. And this, because it has these gigantic phosphate groups, it's very soluble, and it can easily diffuse away from the cell into the cytoplasm. Attached to the membrane, we have a molecule of diacylglycerol, right? Diacyl, so two hydrocarbon tails and a glycerol molecule. This diacylglycerol gets stuck in the membrane, but serves as a docking point for protein kinase C, right? So the alpha subunit of a GQ protein activates the phospholipase C, and it triggers the formation of diacylglycerol, or DAG, and IP3. And the combination of both of these things is what ultimately leads to the changes in cellular behavior. So this is a small table with some of the examples of phospholipase C mediated signaling. You see that acetylcholine talking to smooth muscle causes contraction. Vasopressin in the liver causes breakdown of glycogen. And acetylcholine in the pancreas causes secretion of pancreatic enzymes. The other thing that's important, I mean, the general mechanism by which this pathway becomes activated. The signal binds to the receptor, whatever the signal is. The receptor activates the alpha Q protein, and the alpha Q, the alpha subunit of this GQ protein, will trigger the activation of phospholipase C, and this phospholipase C is bound to the membrane. Phospholipase C will then hydrolyze the linkage, the covalent bond between the inositol group and the diacylglycerol that's stuck on the membrane, and then the inositol triphosphate, or IP3, will be released as a soluble second messenger. The DAG, the diacylglycerol, will stay stuck in the membrane, and it will become a docking site for a protein kinase that's called protein kinase C. Now, the IP3 molecule that has diffused away from the membrane can find its way to its receptor that's called the IP3 receptor. The IP3 receptor is a protein that's on the membrane of the endoplasmic reticulum, where a lot of calcium is stored. And the IP3 receptor is, in fact, a ligand-gated channel, ligand-gated calcium channel. So the binding of IP3 to its receptor triggers the opening of this channel, and it provides a pathway for this calcium, which is highly concentrated in the endoplasmic reticulum, to diffuse into the cytoplasm of the cell. Once this calcium enters the cytosol, a lot of things can happen. One of the things that calcium does, it basically cooperates with diacylglycerol, and it fully activates some versions of PKC. There are some versions of PKC that depend on calcium, some others don't. But the granularity of it is way beyond the scope of this course. Is this something that you need to know? Yes. Any questions? Nope. Okay. Now, one of the things that's actually kind of interesting is that for this, remember that there's an important effect of calcium here. But calcium in the cytoplasm is kept at extremely low concentrations. So just so you get an idea, the extracellular concentration of calcium is roughly 1 millimolar, right? Somewhere between 1 and 2, depending on the tissue, whatever, you can get 1.8 millimolar worth of calcium extracellularly. In the sarcoplasmic reticulum, the concentration of calcium is similar, maybe 1 order of magnitude less. So somewhere between 0.1 millimolar and 1.5 millimolar, 1.8 millimolar, something like that. The cytosolic concentration of calcium is 10 to the negative 7 molar. That means 4 to 5 orders of magnitude less concentrated than in the extracellular environment, right? This means that there's a huge gradient, a huge concentration gradient for calcium between the extracellular fluid and the cytosol, but also between the endoplasmic reticulum and the cytosol. So the cell has to actually spend a significant amount of effort in making sure that the concentration of calcium in the cytosol remains very, 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 very low. In fact, elevated concentrations of calcium, sustained high concentrations of calcium in a cell are toxic. And this basically, this slide here, I'm highlighting three of the mechanisms, the three most important mechanisms that cells use in order to keep calcium in the cell low. There's the Circa pump, which is an active transporter that pumps calcium from the cytosol into the lumen of the endoplasmic reticulum. This is very, very important in muscle cells, in many different cell types, but in muscles specifically. And then on the plasma membrane of the cell, we have two calcium transporters. The first is the calcium pump that the plasma membrane calcium ATPase that directly uses ATP and pumps calcium out. And then there is a secondary active transporter called NCX, or the sodium calcium exchanger. As the name suggests, this transporter basically exchanges sodium for calcium. So these are the three main mechanisms that the cell uses in order to keep calcium in the cytoplasm low. Now, in addition to these transporting elements, which you can see here, right? Calcium is kept low by forcing it to bind to calcium binding molecules. So there's quite a few molecules in the cytoplasm that are able to bind to calcium, and they kind of sequester calcium so that it's not just floating around activating random stuff. So those are the calcium binding molecules. And also there's a relatively high concentration of calcium in the mitochondrial matrix. So there are transporters in the mitochondria that basically they suck up some of the calcium from the cytosol into the mitochondria. And that also helps to maintain cytosolic calcium low. Any questions? Okay. Calcium is, as I mentioned earlier, in and of itself, a second messenger. And the way that calcium works for the most part is by creating waves throughout the cytoplasm. So this is what happens. For instance, at the beginning of every animal life at least, the fertilization of an egg depends on a cytosolic wave of calcium that starts at the place where fertilization takes place. That arrow represents the point on the egg where the sperm will actually fuse. So the fusion of the membrane of the sperm with the membrane of the egg causes an increase in local concentrations of calcium, as you can see from the switch from cool colors to warm colors. And this wave of calcium basically spreads throughout the cytoplasm over, it reaches half of the cell in about 40 seconds. And the way these calcium waves operate is by a kind of a positive feedback that involves the initial opening of some calcium receptors. Say the IP3 receptor triggers calcium exit from the ER, but some of the calcium that triggers some of the calcium that leaves the ER can then open calcium channels nearby. So the stimulus starts here, and then it becomes propagated because the calcium that leaves from one place opens calcium channels on the other, and that's how the system basically propagates itself. Um, the concentration of calcium in the cytosol keeps increasing, and eventually the channels become sensitive to really high concentrations of calcium. So the calcium wave starts, and then it's calcium that helps propagate itself. And then at really high concentrations of calcium, calcium itself blocks or causes the closure of these channels. So you get a wave that propagates, and then it sort of self-terminates, right? So the self-propagation is the positive feedback part. The termination is the negative feedback uh, part. And the integration of these two elements causes what these phenomena that are called calcium waves or oscillations. And the frequency of these oscillations is really important in the regulation of several organs, including the liver, right? Some cells in blood vessels are also regulated by calcium waves and oscillations. In this example is just data from an experiment that you're measuring calcium concentrations over time in liver cells in response to vasopressin, which is a hormone that increases intracellular calcium. And you see that when you put a certain concentration of vasopressin, you get increased, you get waves of calcium. So each one of these peaks represents one calcium wave. And the calcium wave starts and it ends. There's a tiny little time, and then more calcium, and the wave ends repeatedly. And as you increase the concentration of the hormone, you're activating more receptors, and that means that you can increase the frequency of these oscillations. So you see you have more spikes over less periods of time, right? The frequency of these signals ultimately dictates what the cellular outcome is going to be. Does that make sense? Any questions? Okay. Now, calcium, the frequency of calcium oscillations is one thing, but calcium by itself doesn't really do much. Calcium needs to bind to proteins that will eventually do the actual work of changing cellular behavior. So this figure here represents a calmodulin, a protein that I had told you about earlier that serves as a calcium sensor in many cells. So the calmodulin proteins basically has 4 binding sites for calcium. So each molecule of calmodulin can bind 4 calcium ions. When calcium is bound to calmodulin, it can wrap itself around binding partners, and it triggers their activation or their inactivation. An example of calmodulin-mediated responses can be found here. So this on top here, you have the complex of calmodulin bound to calcium. And this calcium calmodulin complex can wrap itself around its target protein, which is a kinase. And this kinase is called CAMKK2. And this kinase serves as it's kind of a master regulator because it can trigger the phosphorylation of 3 other kinases, CAMK4, CAMK1, and AMPK. All of these are different protein kinases that are involved in several different cellular processes. You don't need to know the details of this pathway. All I want you to know is that calcium binding to calmodulin can do this type of work, right? The figure basically also shows what are the most significant physiological effects of activating each of these pathways. AMPK is important because, in particular, because it's sort of an energy sensor in the cell. But the details are not important for the purpose of this class. Now, what I want you to appreciate on this particular graph, you see that these are graphs of kinase activity as a function of time. And what you see here is that when calcium waves or oscillations are at low frequency, you don't really get a lot of CAM kinase activity, right? But if you increase the frequency of calcium oscillations, then the kinase becomes active, and that triggers changes in cellular behavior. Does that make sense? Okay. Now, how do we turn off these GPCR signaling? There's a group of molecules called arrestins, and there's a bunch of other molecules called GRKs or GPCR kinases. So when the ligand binds to the receptor, and the receptor becomes activated and does all of the things that we have discussed so far. One of the things that can become active as a consequence of this pathway is the GPCR kinase. So when the GRK molecule activates, it triggers the phosphorylation of the receptor. It phosphorylates the intracellular side of the receptor. And I hope you can appreciate at this point that when you phosphorylate a protein, quite often you create sites for the binding of other proteins. In this case, the phosphorylated receptor can be recognized by a molecule called arrestin. And arrestin binds to the receptor, and it basically prevents the receptor from interacting with any of its other partners, right? And on top of that, the binding of arrestin actually triggers the endocytosis of the receptor. The receptor can basically be sucked into the cell, put in a vesicle, and removed from the membrane. And the endocytosis receptor is no longer able to sense the presence of the extracellular ligand. Now, GPCRs are one thing, receptor tyrosine kinases are another. These are the type of receptors that we saw as dimers, right? That they dimerize in the presence of the ligand. There's plenty of different types. And in this case, most of these, as you can tell, all of these are growth factors. So these are proteins that are signals that are really important in cellular growth. They control cellular proliferation. They're super important in cell differentiation and such. As you can tell, all of them have in common an intracellular domain that has tyrosine kinase activity. In some cases, the tyrosine kinase domain is a whole chunk of the protein. In other cases, the kinase domain is sort of split, right? And the extracellular side of the receptor may have differerences that give a certain degrees of specificity to the receptor, right? So some of them have like Ig-like domains, some of them have cysteine-rich domains, so on and so forth. It's not necessarily important for you to remember. What I do want you to understand is that despite what happens in the outside, all of them have tyrosine kinase domains intracellularly. The way that they are activated, again, whatever their ligand is, they will basically bind to two monomers, causing the dimerization of the receptor, and this activates the tyrosine kinase domain in the intracellular side. And again, the right monomer will phosphorylate the left phosphorylates the right. Then again, this kind of like a phosphorylation handshake. And then this initial phosphorylation causes further phosphorylation of the receptor. And once the receptor is fully phosphorylated, you get, again, docking sites for binding partners. And the activation of these binding partners by binding to the receptor here is what causes the initiation of downstream signaling processes. Now, importantly, these are examples of the types of proteins that dock on phosphorylated tyrosine kinase receptors. Scientists have been able to map these things pretty detailed for many kinds of receptors. These is the example in particular for the a receptor for PDGF. This is called platelet-derived growth factor receptor. And you see that this is one kind of receptor that has a split tyrosine kinase domain, and the phosphorylated tyrosines at position 740 and 751 serve as docking sites for an enzyme called PI3 kinase. Tyrosine 771 serves as the docking site for GTPase activating protein, and phosphorylated tyrosines at positions 1009 and 1021 serve as docking sites for a different type of phospholipase called phospholipase C gamma. What all of these proteins have in common, these signaling modules have in common, is these SH2 and SH3 domains. These domains, SH2 and SH3, are specific sequences of amino acids that have high affinity for phosphorylated tyrosines. So these are the places in these proteins that can recognize the phosphorylated receptors and mediate their interaction. Does that make sense? Any questions? Okay. And this table basically shows you several types of signals, most of them transcription factors, the receptor family that they bind to, and examples of their responses, right? So for instance, epidermal growth factor EGF binds to a family of EGF receptors. There's plenty of them. But what all of these things, all of these factors basically stimulate cell survival, growth and proliferation, or differentiation, depending on the developmental stage. Insulin binds to the insulin receptor. The insulin receptor is one such receptor tyrosine kinase. The binding of insulin to the insulin receptor stimulates carbohydrate utilization and protein synthesis. There is a growth factor that kind of looks like insulin in a way, but it's a protein. Well, insulin is a protein too. This is called IGF-1, and there's a receptor for it, IGF receptor 1, stimulator of cell growth. IGF-1 is actually the true mediator of the actions of growth hormone, and blah, blah, blah. So, and finally, vascular endothelial growth factor. There are 3 or 4 types of VEGF receptors, and they stimulate several processes that are important for the function of blood vessels. I am going to talk to you about those processes in lecture 12 when we talk about angiogenesis. Now, how do these things work, right? The idea is that the phosphorylated receptor, the phosphorylation sites form docking sites for these adapter proteins with SH2 domains mainly. Sorry. The proteins that can bind to with SH2 domains that can dock on the phosphorylated receptor, they can bind to, those are adapter proteins that can then recruit further proteins such as SOS in this case, which is a guanine exchange factor for a small GTPase called RAS. RAS in its inactive conformation, it's bound to GDP, but when RAS-GF becomes active, it can exchange GDP for GTP, and then RAS becomes activated. Downstream of RAS, what happens is usually the activation of a cascade of kinase proteins. So there is the MAP. MAP stands for mitogen activated protein. So MAP kinase. which is called RAF. The MAP kinase activates MAP kinase, which is also called MEK. MAP kinase activates MAP kinase, which is called ERK. And then MAP kinase does all of the things here that really matter, which is basically changes in it phosphorylates target proteins, changing their activity, or also changing gene expression. This pathway is important. Why is this important? Because several members of this particular pathway, including RAS, SOS, GRAP2, including some of these receptor tyrosine kinases, when they become mutated, they become oncogenes. And when these things are hyperactive, hyperactivity of this pathway leads to uncontrolled cell proliferation, and that's cancer. So if you want to know things about cancer, this is one of the places to start your interest in cancer. Now, not all receptor tyrosine kinases activate or act via a pathway that involves any of that. So in this case, for instance, the activated receptor triggers the docking instead of SOS and GRAP2 and blah, blah, blah that we saw before, we get a kinase called PI3K, right? Phosphoinositide activated kinase, whatever. And this kinase basically triggers the formation of a membrane phospholipid. It phosphorylates a membrane phospholipid that then becomes a docking site for a bunch of other things, including PDK1 and AKT. Those two are important in several different signaling pathways. Most of them are involved in energy management. So in this particular case, PDK1 becomes activated first. PDK1 is a kinase that triggers the phosphorylation of yet another kinase called AKT. AKT can be phosphorylated by a molecule called mTOR. The phosphorylation by mTOR releases active AKT. And then what this does is it prevents apoptosis. So remember, last week we talked a lot about apoptosis and how we regulate that. So this is basically a cell survival signal, right? The activation of AKT phosphorylates BAD. The phosphorylation of BAD basically inactivates it by binding to a protein called 1433. And then it triggers the activation of an anti-apoptotic protein. So again, the RTKs are involved in cell survival, but the activation of these pathways that lead to cell survival depend on these mediators docking onto membrane phospholipids. And that's just a necessary detail. This is just like, that phosphorylated phospholipid, where it comes from. There are several forms of phosphorylation or phosphorylated phospholipids that are important here. Now, to wrap this part up, none of these pathways act on their own, right? The activation of several GPCRs leads to activation of pKa. If you have calcium, you get calcium calmodulin kinase, activation of PKC. But on the other side, receptor tyrosine kinases, you can get direct activation of phospholipase C, leading to activation of CAM kinase or PKC. GRAP2 activating MAP kinases, but this pathway can also activate PI3 kinase and leading to the activation of AKT, so on and so forth. So take home message from this particular slide, everything is connected, right? Activation of one pathway can, if a cell has a mutation in one particular pathway, in some cases, that can be compensated for by upregulating members of a different signaling pathway that ultimately contributes to the ability of the cell to survive and do whatever it is that they need to do. Ultimately, the combination of all of these signaling networks can be boiled down to either the regulation of transcription or the phosphorylation, dephosphorylation, guanine exchange, GTP exchange of different target proteins. And that's ultimately what changes the behavior of the cell. Okay, let's take 10. Sounds good? Because I need to refill my bottle of water. Yes. Okay. So last part, alternative signaling pathways. These are signaling pathways that are important in specific contexts, right? The signaling pathways that we covered in part 2 are generally important almost everywhere. Almost every cell has the ability to respond to some signal using one of those pathways that we covered in part 2. But in part 3, we're going to talk about some specific signaling pathways that are relevant in specific biological contexts. So for that, we are going to There we go. Ignore that. Those are the learning objectives. This basically tells you what the specific pathways are. And we're going to start with the one that's closest to my heart, which is the nitric oxide pathway. Nitric oxide is a gas molecule that can be produced enzymatically by many cells in the body. The importance of nitric oxide was discovered in the 1980s when it was discovered that the relaxation of smooth muscle in blood vessels depended on the production of a gas hormone produced by the endothelium of those cells, of those blood vessels. So in the endothelium, there is a receptor for acetylcholine, which is, again, this signal that can circulate in the bloodstream. It can also be delivered by fibers of the parasympathetic nervous system directly to the endothelium. So this acetylcholine binds to its receptor. The receptor for acetylcholine here is a GQ coupled receptor. So activation of the receptor causes increases in IP3, increases in calcium. Calcium binds to calmodulin, and then calmodulin leads to the activation of an enzyme called nitric oxide synthase. This NO synthase, or NOS, in the endothelium is called endothelial nitric oxide synthase, picks up an amino acid called arginine, and it basically splits arginine into nitric oxide, which then diffuses and has citrulline, which is another amino acid, as a byproduct. Citrulline is not present in proteins, but it is an amino acid. Nitric oxide is a small molecule, and because it's very symmetrical, it's a gas that's also nonpolar. So it diffuses rapidly across the membrane of the endothelial cell, and in blood vessels that are blood vessels that regulate vascular resistance primarily. This nitric oxide diffuses into the neighboring smooth muscle cell. So basically, the endothelium is basically next to the smooth muscle cell, and the nitric oxide binds to the soluble guanylyl cyclase in the cytoplasm of this smooth muscle cell. So when nitric oxide binds to its guanylyl cyclase receptor, the guanylyl cyclase picks up GTP, turns it into cyclic GMP, and then cyclic GMP does a bunch of different things that lead to the relaxation of the muscle, right? So this entire pathway was discovered in the early 1980s, and it was figured out over the next 10 to 15 years. So we ended up we as scientists back then, figured this entire pathway took them about 10 to 15 years to get it right. Now, specifically, those of you in physio will probably see this later on, sort of a veiled thread there. The idea is that the enos that produces nitric oxide in the endothelial cell, so on top you have the endothelial cell, on the bottom you have the smooth muscle cell. And the enos is activated by the calcium calmodulin complex. So actually calmodulin is one of the many factors necessary for this. Activated calmodulin will directly bind to the nitric oxide synthase. But this enzyme actually needs activation by several other components, including calcium and the activation of AKT. So one of these kinases that we were talking about before. The product of this is nitric oxide. Nitric oxide will diffuse into the smooth muscle cell. We get the soluble guanylyl cyclase and production of cyclic GMP. Cyclic GMP will then bind to a kinase protein called protein kinase G. And the protein kinase G will cause the phosphorylation, because it's a kinase, it will cause the phosphorylation of several elements in the smooth muscle cell. It will cause phosphorylation of potassium channels, activating them, and that causes hyperpolarization of the smooth muscle cell, which is associated with relaxation. It inhibits calcium channels, so less calcium in the smooth muscle cell, relaxation. And then it has this dual role in regulating the activity of these two proteins, MLCP, myosin light chain phosphatase, and MLCK, myosin light chain kinase. The kinase is a kinase, so it phosphorylates. The phosphatase is a phosphatase, so it dephosphorylates. And the balance in the levels of activity between these two proteins determines the degree of phosphorylation of myosin in those smooth muscle cells, and that directly affects contraction, right? If you have more MLCP activity, you get less phosphorylation, and that causes relaxation of the smooth muscle. If you have more kinase activity, you have the opposite. More kinase, more phosphorylation, more contraction. So all of the effects of this pathway lead to a decrease in the phosphorylation of myosin and thus the relaxation of the smooth muscle cell. Okay? Those are the players here. Does this make sense? Yep. Okay. Come on, click. There we go. Another relevant pathway is the JAK-STAT pathway, primarily activated by cytokines. The cytokine receptors tend to be associated with these JAK proteins. JAK stands for Janus kinase. So that the inactive form of these receptors, has these JAK proteins sort of already pre-bound to them, but when the cytokine, which is the signal here, when the cytokine binds, the receptors dimerize, and that enables the cross-phosphorylation between the JAK kinases, right? So this is, in a way, this is similar to what happens with the receptor tyrosine kinases. The difference is that these receptors don't have their own kinase activity. They need the JAK for the cross-phosphorylation, right? So it's like they have a separate module that has kinase activity. So the cross-phosphorylation of the JAK kinases then causes the phosphorylation of the receptor, and the phosphorylated receptor serves as a docking site for these proteins called STAT2 and STAT1. And the STAT2 and STAT1, so this is where things are kind of weird. The STAT2 and STAT1 are actually transcription factors, right? but they need to bind to the phosphorylated receptor for them to be phosphorylated by JAK. And JAK activates STAT2 and STAT1, and then they sort of dissociate and they travel to the nucleus. They dimerize like this, and then they ultimately bind to the DNA, form transcription regulation complexes, and that triggers gene expression, right? This is how cytokines, which are, for the most part, we use the word cytokine, particularly when we discuss immunological reactions. So interleukins, tumor necrosis factor, interferons, and a bunch of different things act, among others, via this pathway. So if you are interested in immunology, this is where you start your research. So again, types of factors or signals that operate via the JAK-STAT pathway, interferon gamma and interferon alpha, which are important in the response to viral infections, for instance. Growth hormone uses JAK2, other hormones like erythropoietin, prolactin, and also this GM-CSF, which is super important in the it's a really important factor in the differentiation of cells in the production of immune cells, in other words. So that's basically it. You don't need to memorize it, but understand that this is where these are some of the things that these pathways are involved in. The other thing that I wanted to talk about is TGF-beta. TGF-beta stands for transforming growth factor beta. And there's also another kind of protein called BMPs, bone morphogenetic proteins. These are signaling devices, proteins that are really important in developmental biology. So they actually, they're called morphogens, and they establish gradients so that the relative concentration of these signals determines the cellular fate of proteins as the embryo begins its development. For that reason, they act mostly in a paracrine fashion. So they're produced by signaling cells, and they remain in the interstitial fluid, and at different concentrations, again, they form these gradients. So in the particular case of TGF-beta, the receptor actually tetramerizes, right? So we have basically 2 dimers for a total of 4 receptors. There are, one pair of receptors are called type 2, the other pair is called type B, and the ligand itself, TGF-beta itself, is a dimer. So think of them as receptor tyrosine kinases, but instead of tyrosine kinases, these receptors have serine threonine kinase domains. So I'm sure you've heard that there are 3 amino acids that can be phosphorylated in proteins: serine, threonine, and tyrosine. Those are the 3 amino acids that have a free hydroxyl group in their side chain. Phosphorylation always needs a free hydroxyl group. So tyrosine has one, and the kinases that recognize and phosphorylate tyrosines are called tyrosine kinases. The majority of all the other kinases actually phosphorylate serine or threonines. And this is one such example. In this case, the receptor is a serine threonine kinase. The thing is that for receptors, the majority of them are actually receptor tyrosine kinases. But in this particular case, TGF receptors and BMP receptors act via serine threonine kinase receptors. So the binding of TGF-beta causes the tetramerization of the receptor, and the tetramerized receptor becomes phosphorylated, and it creates binding sites for these regulatory proteins called SMADs, right? And in this case, we're taking a look at SMAD2 and SMAD3. These are receptor-activated SMADs that are recruited by the receptor and then phosphorylated by the receptor. Once they're phosphorylated, SMAD2 and SMAD3, they can bind to another SMAD called SMAD4, and they can move into the nucleus where they form a transcription regulatory complex that basically binds to DNA regions called TGF beta responsive elements, and that triggers transcription. So SMADs are transcription factors that in order to become active, they need to be phosphorylated by the receptor, right? You can think of this and start integrating things from last week. One of the things that probably happens is that the phosphorylation of SMAD2 enables them to bind SMAD4, and the binding of SMAD4 leads to the exposure of nuclear localization signals that then can be recognized by importance so that the entire complex can move into the nucleus and do its job, right? So these are never isolated things. But again, this is the example of TGF beta and BMP. Another relatively important pathway is the Wnt pathway. The Wnt pathway is made by a collection. It's a very large family of proteins that tend to act as paracrine morphogens. So similar to the BMPs and the TGF betas, they establish gradients, and the higher the concentration, the more activation you get, and the degree of activity ultimately determines cell fate. So they also regulate development for that reason. There are two, well, there's actually more than two, but the two canonical pathways are called the beta-catenin and the planar cell polarity pathways. They operate in different ways. This particular figure is highlighting the details of the Wnt beta-catenin pathway. We're not really going to talk about the planar cell polarity pathway. So the elements of the pathway are listed here. On the membrane, there is a receptor, a 7-transmembrane receptor that is sort of a close cousin of the GPCRs. So it's called frizzled, and it's also one of these 7-transmembrane receptors. And it's usually very close by to a co-receptor called LRP. Intracellularly, there is a mediator called disheveled, and there is a relatively large complex of proteins, including a kinase called GSK3, a kinase called CK1, and two regulatory proteins called axin and APC. This complex, APC, axin, GSK3, blah, 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 it triggers the phosphorylation of a protein called beta-catenin, and this basically causes the instability of beta-catenin. And beta-catenin, in the absence of beta-catenin, what we get is the stabilization of an inhibitory group of proteins in the nucleus involving LEF1 and Groucho. I'll tell you in a bit who named these and why these names are so weird. Ultimately, when there is no Wnt signal, all of this means that the target genes of this pathway are turned off, right? So when we put a Wnt signal, we activate frizzled. This activation of frizzled requires LRP, so there is a complex there. And the activated receptor recruits disheveled and triggers the dissociation of this big complex. So this big complex that involves axin and APC, blah, 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 basically can no longer phosphorylate beta-catenin. And now beta-catenin becomes stable. And And the stable beta-catenin can now move into the nucleus and activate the transcription of these target genes. Fun fact, a lot of these genes, and this is not only for the Wnt pathway, but this is true for a bunch of different pathways. They've been genetically identified in the fruit fly. So the way that you identify things in the fruit fly, and again, the fruit fly is a good model because it's easy, it's cheap, it has a short generation time, so you see results in 2 weeks, and they're sensitive. So when you mutate some of these, say you have a mutation in the gene that encodes for frizzled, the mutant fly receives a name that describes the phenotype, right? So in this case, mutation of this receptor probably created weird hair-like structures on the body of the fly, right? And the gene that is mutated receives the name of that phenotype. There's a mutation of a morphogenetic regulator called antennapedia. And as the name suggests, mutation of the gene causes legs to grow where the antennae are supposed to go. And there are several other examples in this. So again, the same thing for disheveled. I'm not sure about Groucho, but again, scientists sometimes, when they discover interesting things, they get naming rights, and then choices are made. So that's basically it. I don't know that Groucho was discovered in the fruit fly, but I can tell you that Wnt was discovered in the fruit fly, and the Wnt is the mammalian homologue of a wing of a gene that when it's mutated in flies causes the flies to not have wings, and thus that the fly gene is called wingless. So anyway, that's the fun fact. This is what happens with the Wnt beta-catenin pathway. What is important here is that besides the details of the who phosphorylates what, the central thing that changes when you put the ligand in is the stabilization of beta-catenin. Beta-catenin is kept away from the nucleus by triggering its proteolysis, right? Its degradation. So activation of the pathway prevents the degradation of beta-catenin, and that ultimately leads to changes in cell behavior. The TNF-alpha pathway is activated in immunological responses and inflammatory responses. Tumor necrosis alpha is tumor necrosis factor alpha is a really important immune mediator. And when TNF-alpha binds to its receptor, what it triggers is the activity of a nuclear transcription factor called NF-kappa B, or nuclear factor kappa B. The way in which this pathway becomes active is a little weird. Again, if you go into immunology in the future, this is what this is a significant part of what your life is going to be. Activation of the receptor, so of TNF-alpha to the TNF-alpha receptor, the receptor trimerizes, and what this does is it activates this complex called IKK, right? And the IKK has a bunch of different elements, including Nemo, blah, blah, blah. Don't forget about that. IKK is more important. So the idea is that the IKK is an inhibitor of kappa B kinase. That's what IKK stands for. So there is this transcription factor called NF-kappa B that, in the absence of TNF-alpha, is associated with I kappa B, which is the inhibitor of kappa B. So this binary complex is inactive. When TNF alpha binds to its receptor, the IKK complex becomes activated, and that triggers the phosphorylation of I kappa B, which you can see here. When I kappa B becomes phosphorylated, then it is a target for ubiquitination, which is the signal that the cell uses to target things for degradation in the proteasome. So, phosphorylation of I kappa B causes its dissociation from NF kappa B. The inhibitor becomes degraded, and the transcription factor, NF kappa B, can be translocated into the nucleus. Again, another example in which the liberation of this inhibitor exposes a nuclear localization signal, and then the transcription factor can bind to the DNA and trigger transcription of genes. So that's how the TNF alpha pathway works. You see that both Wnt beta-catenin and the TNF alpha pathway rely on the controlled degradation of a particular target. The beta-catenin pathway, that degradation is standard, and the activation of the pathway prevents the degradation. In the TNF alpha pathway, the degradation is necessary for the is induced by the pathway, right? So those are similar mechanisms with their own differences. All right. Ultimately, the activity of these pathways depends on the increase in the activity of a particular mediator. Beta-catenin in one example, NF-kappa B in the other. Any questions about this? Okay. Now, I think that this might be one of the last things that we are going to talk about. Yes. So the earlier we started the first part of today talking about contact-dependent signals, right? Contact-dependent signals are important in a bunch of different processes. Primarily for one of the perhaps more evident role for contact-dependent signals is to prevent cells from proliferating once they have filled a particular volume or space, right? In other words, say you decide to donate a piece of your liver because you can donate a piece of your liver, right? And you can donate a piece of your liver because up to a certain point, your liver will regenerate. The liver has a certain capacity for regeneration. Now, if you donate a piece of your liver and you just wait, your liver cells are going to start a proliferating program. They're going to multiply, and they're going to multiply, and they're going to multiply. But then they're just going to reach the point where they're going to fill the space that was left. And they're not going to continue proliferating. If they were to continue proliferating, then that would cause a problem because you have limited room in your abdominal cavity, right? So when these contact inhibit, when that phenomenon of cells stopping from dividing, once they reach their maximum point of contact, is called contact inhibition. And it's one of the main things that prevents tissues from becoming cancerous, right? When contact inhibition is ignored, a cell has the ability to continue proliferating beyond what is normal and necessary, and that's called neoplasia, and that's the basis of many cancers. So this is also important during development. So the idea is that in this particular example, you have developmentally somewhat undifferentiated epithelial cells that are usually derived from the ectoderm in the early development. Now, any of these particular cells here has the potential to become a neural cell, right? However, only one of them will become a neural cell, right? So the idea is that the cell that will specify into neural tissue changes its expression pattern, and it will basically put out a signal called delta. And that signal called delta is that little red thing that's sticking out from its membrane, right? So on the other cells, there are receptors called Notch. And when delta from the soon-to-be neural cell, right? Delta from the soon-to-be neural cell binds to Notch on its neighbors, the neighbors are automatically prevented from also becoming neural cells, right? In other words, this is sort of a cell marking its territory. The cell is saying, by putting delta signal on its membrane, the cell is signaling, hey, I am going to become the brain. The rest of you will follow my orders, right? You cannot become brains as well. So again, the delta is the transmembrane protein that has a signaling domain on the extracellular part, and the receptor is called Notch. So the way that this generates its effect is kind of complex. I'm not really going to ask you all of the details, but the idea here is that Notch is a protein that is synthesized as a transmembrane. Again, this is the receptor, right? The Notch is the receptor. So it's processed in the Golgi, and it has a bunch of cleavage points. And ultimately, what happens is that when the cleaved Notch receptor binds to the delta ligand on the opposite side of the cell, like on the cell across the street, basically, this interaction causes the cleavage of the entire rest of the separation, really, of the Notch receptor. In this entire complex, the delta with the extracellular Notch domain gets endocytosed by this cell here. And then the remaining section of the Notch receptor triggers another cleavage event, basically, where the intracellular domain of the receptor, so Notch here, migrates to the nucleus, and it turns into a transcription factor. And this transcription factor basically triggers the expression of genes that, when all of this is said and done, prevents this cell here from becoming anything close to the cell that has the delta signal, right? So that's sort of how this pathway works. Again, this is one of the perhaps the best understood way of establishing lateral inhibition. This is a particular case of a protein that's important during development, but there are other forms of contact inhibition at different places. But that's it. That's all I have for today. Thank you for your attention. If you come up with questions, please don't hesitate to email me. We can talk about it at any time. In the meantime, get home safe. Take care.
Thank you, Dr. Mojica. Have a good night.
Thank you. And I'll see you in a few weeks, actually.